Assalamu alaikum. Good evening, everyone, and we welcome to tonight's engaging webinar on crafting compelling research, mastering question framing, aims, and objectives. I am Amna Noor, a community pharmacist, a research scholar, and an executive member of International Biomedical Research Community. I am excited to guide you through this session hosted by RxConnectPk. Before we start today's session, I have a few important reminders. Your engagement is essential to us and we appreciate it. Please fill out the feedback form we will share shortly. Your input is invaluable as we strive to improve our future sessions. If you would like to receive an e-certificate for our attendance, please mark your attendance. The link will be shared in the live chat. We kindly ask that all participants refrain from promotional content during the webinar to maintain focus on our educational objectives. Now let me share a brief introduction about RxConnectPk. This is our 16th webinar and we are proud to have received over 15,000 feedback responses and amassed 20,000 views. Our mission is to empower, connect, and heal the pharmacy community through education, advocacy, and digital engagement. We are dedicated to providing essential knowledge, job opportunities, and networking while promoting medical awareness initiatives. Our focus on empowerment, collaboration, and excellence drives our commitment to creating positive change in healthcare. Tonight, we have the privilege of hearing from Dr. Sheraz Ahmed Khan, a leading expert in global health. With an impressive academic background that includes an MBBS, an MS, and a PhD in global health from the University of Edinburgh, Dr. Shiraz currently serves as the Dean of Global Health at the Health Services Academy and has collaborated with development banks and UN agencies as a senior consultant. With over 24 publications to his name, Dr. Shiraz has made significant contributions to public health and global health. His expertise extends beyond academia as he has actively participated in meaningful projects with leading global organizations. Tonight, he will share his invaluable insights on crafting compelling research, equipping us with the tools we need to excel. Dr. Shiraz, we are all eager to learn from you. Let's give him our full attention. Over to you, Dr. Shiraz Ahmed.
Thank you very much, Amna. It's really a pleasure being here among so many competent and bright people, and who have the energy to attend a webinar on a Monday evening. Monday is one of the most difficult days in the week. So I'll try to be brief and I'll try to be collaborative to the extent that I can be. Uh, the moderators, I ask a simple question. Okay, first of all, please let me know, are there any international audience जिनको उर्दू उर्दू समझ नहीं आएगी?
Sir, almost all is national audience. No one is in.
National audience. Okay. So I'll just keep it very interactive, and आप मुझे बताइएगा because you are looking at the chat box. How many of you are planning to have an MPhil or a PhD? All those who are planning to have an MPhil or a PhD, raise your hands or drop your messages or whatever mode of expression you have in this platform. Okay, so are these responses going to be on your YouTube channel?
Yes, sir.
Okay, so are you getting any responses?
Yes, we have lots of responses, and majority are planning to pursue education in MPhil and for PhD.
Okay, just out of curiosity, can I also look at the YouTube channel and see through the comments?
So you can click on the comments box. It's showing on your screen.
Okay. Okay. Okay, Mohammad Yusuf from Bangladesh. I'm good, Yusuf. Thank you very much. MPhil Pharmacology, Tehreem is planning it, Noyana is planning it, Manahil is planning for MPhil. Okay, Anila applied for MPhil, Madiha Amir Baksh uh, from, okay, yes sir, me, Alize, Manahil. Okay, video diary is saying I'm doing right now and trying to decide my topic of research. Anila Yunus. Okay, so can video diaries? You might have some research ideas already. Can you just share what is your research question and what are your research topics so that we make this webinar very interactive and I do not give a lecture. That's usually not my style. And maybe towards the end of this webinar, at least one person will have a question and very good objectives. Okay, do we have any answer?
Yes, sir. We have one, and she's doing MPhil in pharmacognosy, and she's trying to decide plant having activity of wound healing and analgesic properties. She just give a random idea on his topic, her topic, not
Okay, to decide plant having activity of wound healing and
yes,
analgesic
analgesic.
property. Analgesic property. Okay, that's really nice. So, how would you phrase your question? You are doing MPhil and you want to, okay, no planning. No planning is the best part. MPhil in आप करना चाह रहे हैं and you want to have some research on a plant and see its wound healing and analgesic property. So how will you phrase your question? Okay, so my apologies, guys. I thought it will be a live interactive session, and it's only a chat, so it is not as interactive as I wanted it to be. Uh, in that case, maybe I'll just go through uh, the core concepts that I wanted to conduct through an interactive session. You know, okay. So for a good research, if you are planning to have an MPhil or a PhD, you need to have a very good research question. And a research question, how do you arrive at your research question? So if you are going to work on a plant and you want to see its analgesic properties or you want to see its wound healing properties, you first need to have a thorough literature review. if somebody else has ever conducted research on this plant. If a research has been conducted on this plant, then you need to answer whether you uh, are required to do or replicate a research that is already done. And there is usually no point in repeating a research that is already done. Then you need to phrase your question in a very methodical way so that you come up with the real outcome of your research. And the best research question should have some elements. Uh, One element is that you should have a specific and defined population for your research. So the population or the patient group needs to be very clearly defined. The question, the acronym used for a question is PICOT, that is PICOT. So the population needs to be very, very effective. Now, if you want to have the study of the analgesic property and the healing properties of a plant, you need to tell me in which population do you want to do it. For example, there may be an acute wound and you want to have to check its properties, or do you want to have uh, uh, to test it with an altogether different population? For example, somebody having diabetic foot ulcers. Now that is a wound, but there is entirely separate thing, you know, because there are a lot of things going into the healing process, and it is not only the chemical or the medicine that you are giving, but the underlying pathophysiology and the underlying blood supply and everything. So if you do not define your population uh, in a very methodical manner, then your results may not be very rigorous, right? By population or the patient group, you need to be very specific about the age, gender, sometimes ethnicity, and specific health issues. For example, if you are testing uh, antihypertensive, we already know that the black ethnic groups and African descent people, they respond differently to antihypertensive than the Caucasian people, you know. The second thing is that you need to define your intervention very, very clearly. So, what is the main intervention that you are considering? Here, you are considering testing the effects of certain drugs or its derivative, right? So, this is a treatment that you are going to test. Another person would like to test a diagnostic kit. Somebody would like to test a prevention strategy. Now, can you apply this medicine or its extract on early wound, stage one wound, to prevent its progression? That is one type of intervention. Or do you want to use it in an advanced and deep ulcer? There is another type of intervention. So, if you use it across a variety of ulcers, or if you are doing a variety of interventions, that is using this plant for preventive purposes as well as for the treatment purposes, then you will have to configure your research methodology according to your intervention. So once you have determined your population and your intervention, then you need to have a comparison or a control group, right? Now, for example, if you decide to test this in chronic wounds, right? And your interest is in diabetic patients, and you are applying it to diabetic patients, but you do not have any control group, how can you say whether this works better than a control group? So, is there a comparison intervention or a control group? This could be another treatment or a placebo or no intervention at all. So, if you take 500 diabetes mellitus patients who have advanced wounds, and you kind of divide them into two groups blindly, and you give the standard therapy, which is given for the wound healing. For example, you wash the wound with normal saline and you apply dry dressing. And if it is infected, then you also need to apply, at times, wash it with antiseptic and then dry dressing and continue with systemic antibiotic. This is the standard regime. And if you want to test these new drugs, so 50% of the people will have the standard regime, and the remaining 50% of the people will have the standard regime plus the intervention. That is the plant from which you have the extract, and you apply it at some point in time. For example, after a thorough wash of the wound, you and antiseptic, you use the same you followed the same protocol for both, but in this case you have applied the extract from that plant as well. And then you follow the patient for 2 weeks or 3 weeks or the standard therapy time, and then you look at the wound and score the wound in both the groups, the control group and the intervention group. So once you have the population, that is diabetic population with foot ulcer, and the intervention is your plant derivative, the control is standard therapy. Now you look at the outcome. What is the outcome at which you are looking? So number one outcome is visual inspection. So you have a score of, let's say, how deep the wound was. Was it a stage 4 ulcer? That is, the wound had exposed the bone as well, the bone, or was it stage 2, in which 3, in which the infection or the wound had exposed the muscle but not the bone, or was it stage 2, in which the skin was infected or the skin was affected but the muscle was not exposed, or was it stage 1? Stage 1, where you have only the involvement of the epidermis and all the remaining things are... Hello. See this lady. She has been looking for her papa. Papa is here, okay? Okay. So if you have defined your population, your intervention, and your comparison, then you also need to define your outcome. Now, on a visual inspection, you can go for whether stage 4 wound has converted into 3, 3 has converted into 2, 2 has converted into 1, or if there is no wound at all. So that is your outcome. And then you have to define your time, or what time frame is the outcome expected. This could refer to the duration of the treatment or follow-up period or time to observe the effect. Now, if you have a question that is having all these components, then you have a very, very good quality research question. Now, if your question is, sorry, moderator, can I go to the YouTube, or will it disconnect me from here?
You can open this link. You web.
Okay. Okay, thank you very much. So maybe I can now look at the joint conversation. Okay, so let's say the medicine or the intervention, the medicine that you have, the molecules that you have extracted, we call it treatment A, TK, and the control group where you are giving only the control therapy is treatment B, ठीक है. Now, if we want to have a PICOT question is, uh,
Excuse me,
does
sir.
Mhm.
Just for 1 second. Sir, audience is saying please continue in the Urdu. It will be more understandable for them. तो अगर
उर्दू में
उर्दू में आप
कंटिन्यू करूँ?
request करते हैं. Okay, yes.
उर्दू में कंटिन्यू करूँ?
Yes, sir.
Okay. So, but there was one colleague from Bangladesh. Can he understand Urdu?
Yes, I guess he can understand.
Okay, perfect. So, अगर आप ट्रीटमेंट ए और ट्रीटमेंट बी की कंपैरिजन करना चाहते हैं, सो पॉपुलेशन मैंने बताया कि डायबिटिक पेशेंट्स विद फुट अल्सर्स, ठीक है. सो पी कॉट का पी हमने कवर कर दिया. पी इज डायबिटिक पेशेंट विद फुट अल्सर, ठीक है. पी के बाद आई है, दैट इज इंटरवेंशन. व्हाट इज द इंटरवेंशन? सो इंटरवेंशंस आर treatment A, in which ये जो colleague बता रहे थे कि he is interested in doing some plant-based research, उसमें से जो आपका extract है, that is treatment A. And what is the comparator or control group? That is treatment B. Treatment B is standard wound care, ठीक है. What is the outcome? So outcome पे मैं कहूँगा कि wound healing स्कोर या ग्रेडिंग, ठीक है. कि आपने विजुअल इंस्पेक्शन किया और आपने उसकी ग्रेडिंग की. एक तरीका ये हो सकता है. दूसरा तरीका ये हो सकता है कि क्रॉनिक वूंड्स के अंदर ईएसआर जो है, क्रॉनिक इन्फेक्शन क्रॉनिक वूंड्स के अंदर ईएसआर, एरिथ्रोसाइट सेडिमेंटेशन रेट, वो इंक्रीज हो जाता है. एंड दूसरा जो इन्फ्लेमेटरी मार्कर्स हैं, इन्फ्लेमेटरी मार्कर्स आपके इंक्रीज हो जाते हैं, ठीक है. सो अगर आप विजुअल इंस्पेक्शन तक रखते हैं तो आप विजुअल इंस्पेक्शन तक रखें. लेकिन अगर आप आउटकम बेस्ड ऑन लैब्स करना चाहते हैं तो उसमें आप डिफरेंट इन्फ्लेमेटरी मार्कर्स और ईएसआर को भी ऐड कर सकते हैं, ठीक है. एंड टाइम ड्यूरेशन जो है दैट डिपेंड्स ऑन कि जब आप अपना ट्रायल करेंगे उसमें आप डिटरमिन करेंगे कि कितने टाइम ड्यूरेशन के लिए आप इसको कंटिन्यू करना चाहेंगे. फॉर इंस्टेंस जब हमारे पास पेशेंट्स आते थे, दे वुड यूजुअली स्टे बिटवीन 14 days to 21 days, either डायबिटोलॉजी एंडोक्राइनोलॉजी वार्ड में 2 हफ्ते की ट्रीटमेंट लेते थे या 3 हफ्ते की ट्रीटमेंट लेते थे, ठीक है. एंड उनकी रेगुलर ड्रेसिंग और वो करवाई जाती थी. सो लेट्स से हम इसको लिख लेते हैं 2 weeks, ठीक है. नाउ द क्वेश्चन वुड बी, आर द outcomes for chronic foot ulcer, are the outcome for chronic foot ulcer different in diabetic patients kept on treatment A versus treatment B, ठीक है. सो आर द आउटकम्स, आउटकम की मैंने बात की. आउटकम क्या है? वूंड हीलिंग. सो आर द वूंड healing, or let's say, let's slightly rephrase it, is wound healing for chronic foot ulcer different in diabetic patients kept on treatment A versus treatment B administered over 2 weeks? अब आप मुझे ये बताएं चैट बॉक्स में कि कैन यू आइडेंटिफाई द पॉपुलेशन, द कंपैरेटर, द इंटरवेंशन, द आउटकम एंड द टाइम इन दिस क्वेश्चन. मॉडरेटर्स मुझे आप बताते जाएं कि हाउ मेनी पीपल थिंक कैन दे कैन आइडेंटिफाई all five elements in this one question.
Sure, sir. Sir, it would be great if you can share your screen so that audience can engage more with the guide.
ओके, मैं क्या करूं, मुझे गाइड कीजिएगा.
सर आप सर आप स्क्रीन शेयर कर देंगे तो उससे ऑडियंस ज़्यादा इंगेज करेगी.
Okay, and how do I share my screen?
Aisa, can you please guide sir about how we can share screen? Sir, you can go to present. There is option visible on the screen about present.
Okay, let me go to it. Actually, apologies, I don't know, I have never been on this platform. I prefer Zoom or Blackboard Collaborate. This is my first time on this platform, so please bear with me. And I thought we will be going on Zoom. Uh, uh, uh. Okay.
सर आपको प्रेजेंट का साइन शो हो रहा होगा.
मैं देख रहा हूँ, पहले मैं ज़रा इस डॉक्यूमेंट को शेप में करूँ एंड देन मैं आपको बताता हूँ. प्रेजेंट का ऑप्शन, यस,
ये सर उस पर क्लिक करें, स्लाइड्स
शेयर
पर
स्क्रीन.
जाकर. यस सर,
I'm sharing my
जितना
screen.
आपके कंप्यूटर में होंगे उसको जाके अपलोड कर दें.
Okay, so I'm just sharing my screen. Home tab, window, entire screen. I'll go for entire screen. Entire screen share नहीं हो पा रहा. Okay, RxConnect webinar. Okay, can you see my screen now? Moderators, can you see my screen?
Yes, sir.
ओके, सो देखिए, हमने कहा कि पहले आप पॉपुलेशन को आइडेंटिफाई करें. कैन यू सी पॉपुलेशन इन दिस ग्रुप? मॉडरेटर्स, चले आप ही इंटरेक्ट करें मेरे साथ ऑन बिहाफ ऑफ द ग्रुप. वेयर इज द पॉपुलेशन?
Sure, sir. So diabetic patients is the population in this. Sir,
ओके,
as I am a research scholar, so I will be engaging with the as an audience. Sir, and food
you
ulcer
you
is my interest of topic.
you are also a research scholar.
Yes, sir.
Okay, best, you can speak on their behalf then. Okay,
Sure, sir.
so let's say this is the population. Not let's say, indeed it is the population. Okay, give me the intervention. Where is the intervention?
Intervention is the treatment A and treatment B. So intervention is
Treatment
the
A is the intervention, जो प्लांट एक्सट्रैक्ट जिस पे हमारे क्लिक काम करना चाहते हैं, उनकी उसकी हीलिंग प्रॉपर्टीज देखना चाहते हैं. एंड वेयर इज द कंट्रोल ग्रुप और द कंपैरेटर?
treatment B.
ट्रीटमेंट बी. ट्रीटमेंट बी में हमने क्या कहा था कि क्या इंक्लूडेड है?
It's just placebo and or any traditional good.
No, it's not placebo. It is treatment as usual.
Regular
In
treatment उसमें use कर
या
लेते हैं.
या स्टैंडर्ड ट्रीटमेंट जो मैंने बताया, नॉर्मल सलाइन के साथ वॉश किया, पायोडीन के साथ साफ किया, फिर सलाइन के साथ वॉश किया, फिर ड्राई ड्रेसिंग अप्लाई की और एंटीबायोटिक जारी रखे. दिस इज द स्टैंडर्ड ट्रीटमेंट. ठीक है. ओके, ट्रीटमेंट भी हो गई. एंड आउटकम क्या है?
Wound healing of chronic food ulcer is the outcome.
आउटकम है. ओके, लास्ट क्या रहता है? टाइम.
Time
टाइम हमने डिसाइड किया
2 weeks.
2 weeks. Now, is this a PICOT question?
Yes, absolutely.
Have we missed anything? Have we missed anything?
No, sir.
No. So we defined the population very neat and clean, diabetic patients having foot ulcers. And we decided the intervention, that is giving the plant extract, which is treatment A. Then we decided the comparator or the control group. The comparator is treatment B, or the control group is that group who are receiving treatment B. The outcome we decided is wound healing. वूंड हीलिंग को आप ऑपरेशनलाइज करेंगे कि आप वूंड हीलिंग स्कोर कैसे करते हैं, या विजुअल इंस्पेक्शन से करेंगे, या आप सीरोलॉजी से या हेमाटोलॉजिकल जो आपके मार्कर्स हैं उससे आप करेंगे, ब्लड मार्कर से करेंगे. सो देयर आर सम इन्फ्लेमेटरी मार्कर्स कि आप डेली बेसिस पे दोनों ट्रीटमेंट एडमिनिस्टर कर रहे हैं, साथ साथ आप उस सीरम में इन्फ्लेमेटरी मार्कर्स को भी देख रहे हैं. ठीक है जी. And you do it for 2 weeks. 2 weeks के बाद you will have your results. So अगर आपने उसमें किया है, सिर्फ visual inspection पे किया है, you will have the counts, the number of people in treatment A and treatment B who have regressed from stage 4 ulcer to stage 1 ulcer. ठीक है? So that will be pretty straightforward conclusion. And then you will look at treatment B, और उसमें देखेंगे the number of patients who have downgraded from stage 4 to stage 1, and you will have your answer, and based on that answer you will see जो statistical test आप apply करेंगे, जो कि आज हमारे इस topic से ज़्यादा advance है, इसमें cover नहीं करेंगे, but through a statistical analysis there will be some differences. But are those differences by chance, or are those differences really due to your intervention? To इसको define करने के लिए you will have to undertake some statistical tests, और उन statistical tests पे आप p value को देखेंगे. अगर p value is significant, you will say कि treatment A is superior to treatment B. अगर p value significant नहीं है and you have the same values, you will say the treatment A is equivalent to treatment B, or you can say treatment A is inferior to treatment B. So these are the type of outcomes that you will determine towards the end of your clinical trial. So we have discussed a PICOT question, right? Now the second thing that you need to understand is that जब आप एक रिसर्च करते हैं तो उसके ऑब्जेक्टिव्स जब आप रखते हैं वो फाइनर के क्राइटेरिया पे पूरे होने चाहिए. फाइनर में F stands for feasible, I stands for interesting, N stands for novel, E stands for ethical, R stands for relevant. ठीक है. अभी जो आपने प्लांट एक्सट्रैक्ट की बात की, is this feasible? If you have the laboratory and if you have the diabetes patients with foot ulcers, and if you have the facilities to offer them the standard therapy plus this adds on, then it is feasible. Do you have sufficient number of diabetic foot ulcers के उसमें randomize करके आप 2 groups बनाएं? अगर हैं तो yes, then it is feasible. Is it interesting? So it is definitely interesting because diabetes में foot ulcer is the most debilitating chronic complications. You know, it is one of the longest, I would say, and earliest and longest problem. So if you are proposing to find a cure towards it, it is definitely interesting to the patient community, and it is definitely interesting for the scientific community. Number 3, it should be novel. ठीक है. Novel क्या हो कि if you are repeating the same thing with the same group of people in the same locality, then there is no need to do it, right? But if you are treating, if you are working on a plant or an extract जिस पे पहले किसी ने काम नहीं किया, then it is novel. If you are working on a technology जिस पे पहले किसी ने काम नहीं किया, then it is novel. For example, we saw in the COVID-19 that Astra के जो Pfizer थे और Moderna जो थे, they went for messenger RNA vaccine, the first of its kind. There were no vaccines based on messenger RNA. So it was an absolutely novel approach. And due to that novel approach, they had an efficacy of 95%. कंपेयर टू इट जो स्टैंडर्ड थेरेपीज थी कि आप एंटीजन लें और एंटीजन को आप उसकी भी जो इन्फेक्टिविटी है वो उसको आप डिक्रीज करें, एटन्यूएट करें, फिर आप इंट्रोड्यूस करें बॉडी में और फिर उसके खिलाफ इम्यून सिस्टम एक्टिवेट हो. देयर वाज अ स्टैंडर्ड अप्रोच. ठीक है, उसमें कुछ वैक्सीन ऐसी थी कि जिसकी 52% एफिकेसी थी, कुछ की 53% थी और कुछ की 70 के रेंज में थी. ठीक है, बट द मोस्ट नोवेल अप्रोच वाज दैट ऑफ द मैसेंजर आरएनए. राइट, अब क्या मैसेंजर आरएनए के ऊपर जो रिसर्च हुई वाज दैट एथिकल एज वेल. राइट, so to be ethical there are certain principles. Number one is do no harm. ऐसी थेरेपी दो कि जिससे हार्म ना हो. फायदा होता है नहीं होता, लेकिन द फर्स्ट प्रिंसिपल इज कि उससे हार्म ना हो. ठीक है, so if you are giving a messenger RNA to produce a certain protein that can kill a certain virus, that is definitely a good proposition. There is a potential for benefit. But can that messenger RNA reproduce or produce any harmful protein as well? So first you need to test that in a laboratory environment if it is producing any harmful protein as well. If it is producing a harmful protein, or if it is producing uncontrollable replication of proteins, then it is a no-go. This is novel, but this is unethical. So फिर आप इसको नहीं करेंगे. ठीक है, but the thing is कि if the novel strategy that you are applying does not have any known adverse effect, you cannot hypothesize about any potential harm to the patient. And if you think if there is some harm, जिस तरह के एस्ट्राजेनेका के वैक्सीन के साथ मायोकार्डाइटिस हुआ था, but the harm outweighs the benefit outweighs the harm. For example, अगर at the age of 70 किसी को कोरोना हो जाए, so the death was almost certain initially जब treatment protocols develop नहीं हुए थे. लेकिन myocarditis के साथ a person can live with myocarditis for quite some time, you know. If you are taking a life-saving medicine, but like chemotherapeutics, but the adverse effect is that you will have a hair loss. Now, a hair loss is something with which one can live, and people take this risk in order to save their life. Therefore, if you are producing something where the benefit outweighs the risk, then again it is relevant, it is ethical. Sorry for the disturbance. Okay? And the last thing is it relevant? Okay? So the question must be relevant to the field of study, public health, clinical practice, or the society. It should contribute meaningfully to the body of knowledge or address important health or societal issues. Okay? Does the question address a significant issue in healthcare or improve patient outcome? Right? Now, if you look at the same thing, relevance, if say you are producing a vaccine essentially for the same COVID-19. Now, COVID-19 is having vaccines that are 95% effective, right? But there is no vaccine which is 95% effective for Ebola. Now, if there are two questions, if you are a researcher and you want to propose a research, then it should better be, and you are in vaccine manufacturing, then your question should better be on Ebola vaccine rather than COVID-19 vaccine, because COVID-19 vaccine, they are having an inventory of vaccines now which are quite safe and efficacious, as well as readily available in today's market. So relevance to the medical field and relevance to the society or the patient is another thing that you need to keep in mind. So if you write a question, there is PICOT, and if you write your research objectives that are feasible, interesting, novel, ethical, and relevant, then you can have a very, very good research which will have a very positive impact on the outcome of the health of the patients, or if it is for prevention, for it will have a very good outcome on preventing the population against a certain disease, right? So I'll stop here. It was just a brief introduction into how to phrase a question and how to write a finer uh, objectives. Uh, the Maybe I can take a few questions if the moderators can present a few questions. But if there is interest, I don't know how many people are attending the session right now, and how many people think that it was helpful to them. So the moderators, maybe if you can run a poll and ask the participants about how did they find this session, or whether they would like to have a continuation of this session, then maybe we can run a full course on research methodology. So from objectives, then we can go towards the question and objectives. We can go towards the methodologies. We can define the variables. Then we can define the sampling size. Then we can define the study design. And then we can go towards the methodological approaches and everything. So I'll stop here. And if there are any questions, I'm happy to take your questions in the remaining time. Should I stop my screen sharing now, Amna?
Yes, sir.
Okay. Okay, thank you. I did it.
Okay, sir. We are waiting from audience for the response. Sir, our attendees has found it helpful, so I guess we should continue with the training itself.
Very insightful. Asif Nawaz, helpful. Thank you, Asif. Rufia Khan, helpful for me. It was very well and informative. Madiha, thank you. Amna, please write your feedback. Okay, that's all, Amna. Sibghatullah, we need more of this kind. Okay, Afia, it was such a helpful session. Thanks for elaborating it in detail. Your way of explanation is too much similar to our pharmacology teacher, Professor Hakim Mehman. Okay, I'm glad. So, Muhammad Zaid, helpful. Nayana, very informative. Sir, these both objectives applicable for every study design or for a specific. So Chaudhry Shagil Saab, this is for every study. So the same principles apply for social sciences, the same principles apply for medical sciences, pharmacology, you name it, and these will apply. Okay, so no problem. Natural health and beauty, what strategies do you recommend for formulating research questions specifically related to drug efficacy and safety? Natural health and beauty, maybe you can go through the video which the moderators will share with you. We have exactly answered this question during this webinar. Can you please repeat statistical analysis? You were talking about p-value significance. I missed that part. No video diaries. मैंने कहा था कि अगर interest है तो फिर हम मज़ीद webinars कर सकते हैं and उसमें statistical analysis और उन चीज़ों के बारे में भी हम बात कर सकते हैं. How to finalize a topic for research? Well, it depends upon you. Topic if you define the question, that becomes your topic. Okay, you're doing great. It is helpful, not in gaining knowledge. Okay, thank you. Accession review article पे भी रख दें. Okay, we can do that. Any tips for formulating a questionnaire for research? Questionnaire पे I think मुझे 2-3 सेशन लेने पड़ेंगे. एक सेशन मुझे क्वेश्चनियर के फेस वैलिडिटी पे लेना पड़ेगा, एक उसके कंटेंट वैलिडिटी पे लेना पड़ेगा और एक फिर उसके वैलिडेशन और पायलटिंग पे लेना पड़ेगा. So the tip is for your questionnaire, it should be face valid, it should be content valid, and it should be piloted. External validity, internal validity. What are some common challenges pharmacists encounter when framing research question, and how can these challenges be overcome? Well, I think that the challenges are the same. If you are not clear about your population, intervention, control group outcomes and time, or pharmacist or doctor or nurse, so their research will be a waste of time. And if your objectives are not insightful, not feasible, not interesting, not novel, not ethical, it will be a bad research. Be it an astrophysicist or a pharmacist, applies to both. It has always been hard selecting the right research topic or question. Can you please elaborate that? Well, look into your heart and see what inspires you. If you think you can make a difference in cancer care and discovering a cure for cancer, go for it. If you think you are more like a public health guy and you want to do pharmacovigilance, go for pharmacovigilance kind of questions. So it is entirely your own choice. How can pharmacists navigate ethical consideration when framing research questions that involve vulnerable population, particularly in clinical trials and observation studies? So there are standards for ethical research, and I can share some content with the moderators and they can share it with you. And basic principle is do no harm, try to make it useful to them. Number 3, informed and ethical consent. Number 4, जो आपके वो है, the subject should have full autonomy and the data should be confidential. These are basic tenets. If you have these things in place, you are a very good researcher and your research is ethical. Guide how to register our research work in DREP when we select novel topics. Well, since I'm not a pharmacist by background, maybe some colleagues in the groups, senior pharmacists, they can address this question, how to register with DREP. One last question from my side. Yes, natural. What is it? In your experience, how can ensure that the research aims are not only relevant but also actionable within clinical practice, particularly in areas where medication adherence and safety monitoring? Well, this is a very broad topic. So if you have a good product but people are self-medicating, then it is of no use. If you have a novel therapy but people cannot afford it, it is of no use. So these are broader health system level questions rather than more clinical or laboratory related questions. And pharmacoeconomics is my favorite topic of research. So pharmacoeconomics, if somebody is interested, we can talk at length about pharmacoeconomics. Nestle, I just summarized those documents to you. Do no harm, make it beneficial, informed consent, data privacy, and their ability to withdraw their consent at any point. You ensure these things and your research is safe, you are ethical. We should also have a session on how to find research gaps or question. Yes, that comes under the ambit of literature review. Literature review in itself is an art. Yesterday, I attended a workshop over here, and that was talking about searching different databases. But for literature review and identifying the gap, it can be a standalone webinar itself. Can you arrange another session through RxConnectPk? Yes, of course, we can, but next time I would request him to make it on Zoom, because it feels like I'm going back into the uh, AOL or MSN chat era, which we left behind 20 years ago. Okay, sir, thank you. But I really appreciate RX Connect bringing so many professionals and making knowledge so accessible. Okay, at night, after your duties and after everything, you can have session like these. A lot of interest topic of research question, a lot of limitations. Lack of patient. Yes, that's true. There was a good session information. Sir, we are more comfortable with YouTube as a youth. Okay, so maybe generation gap. I'm more comfortable with. I mean, YouTube is fine coming to you by YouTube, but the problem is I cannot hear you, and communication is complete when it is two-way. So maybe you could have asked many good questions. You could have contributed very productively. That is what I'm missing. So it is honestly. You might know a lot of things जो कि मुझे नहीं पता होंगी, and I would love to learn from you folks as well. So, okay, I'll stop here. Maybe I have gone a bit over the top, and my host is
Thank you so much,
host.
Dr. Shiraz, for your profound insights and taking the time to answer our questions. So your expertise has enriched our understanding and equipped us with actionable strategies to elevate our research. I personally have learned a lot, and it helped me refine my final, especially final statement as a research scholar. So a quick
Perfect.
note for everyone, kindly please fill out the feedback form to receive your e-certificates for the today's session. At RxConnectPk, we remain committed to empowering the pharmacy community through educational initiatives like this one. We look forward to seeing you at our future events too. So thank you all for joining us and have a wonderful evening. Thank you so much, Dr. Shiraz,
Thank,
today.
thank you very much. Allah Hafiz.
Allah Hafiz.